We're going to take a look at two mechanisms in a bit of detail, the first of those being chymotrypsin. Chymotrypsin is in the class of enzymes called the serine proteases, and they are so named because they have a serine in the active site that participates in the hydrolysis of peptide bonds, thus the protease part of it. And the active site contains what's called a catalytic triad, which is composed first of all of the serine residue, a histidine residue that participates in acid-base reactions, and an aspartic acid residue that, with its negative charge here, it pulls on this proton and it makes histidine a better base than it normally would be. In fact, if we take that aspartic acid residue away, histidine is not strong enough to deprotonate the serine, because we need the serine in a deprotonated state, because this is what's going to attack the carbonyl of our peptide bond. The serine proteases, there are a variety of these. Chymotrypsin specifically uses aromatic amino acids as the substrate. If we look at the substrate for chymotrypsin, here's phenylalanine, and we cleave this bond right there on the carboxyl side of phenylalanine. And the reason for this is that the binding site for chymotrypsin has this hydrophobic pocket that nicely accommodates things like phenylalanine or tryptophan. It doesn't accommodate the polar amino acids, although it does accommodate tyrosine. It doesn't accommodate things like serine, because serine flops around too much in that site. Same with alanine. Alanine has a relatively small R group. Although it's hydrophobic, it doesn't form the interactions that are needed to get the right orientation for catalysis to occur. There are a variety of serine proteases that have other binding sites that make them specific for other amino acids. And simply by changing that binding site but keeping the same catalytic triad, we can get several different specificities in terms of the substrate that we can use, but have the same mechanism, because the mechanism applies to the breaking of any peptide bond. It doesn't have to be just those that have an aromatic amino acid at that position. So we start our mechanism with our peptide, and we'll notice here that we've got R1 and R2, and R1 is the inside of the peptide bond. Normally we think of, we write peptides from N to C. So in this case we have R1 R2 like that. It's reversed in this figure just because this is most convenient for how the enzyme is organized. So the first step is our peptide bond. Our peptide binds to the enzyme, and we need to deprotonate our serine residue such that it can perform this nucleophilic attack on that carbonyl. And this is done by histidine deprotonating the serine residue and generating an O-, which then attacks the carbonyl. These electrons in the carbon-oxygen double bond here are going to move up onto the oxygen, and we've got this negative charge there. That is stabilized by having, for example, a lysine residue with its positive charge adjacent to that oxygen to attract that negative charge that's going to form in that oxygen of the carbonyl. This is our first tetrahedral intermediate that we form. And tetrahedral intermediates around carbon, they tend to break down somewhat, and that's what we ultimately want to do, because we're going to do that by breaking the nitrogen to carbon bond. And that's going to generate a new amine, and the carboxylic acid portion is still going to be bound to the enzyme. So at this point, our amine forms. The amine is going to remove the proton from histidine that we see here, and it's going to leave the enzyme as the newly formed amine group. Now we still have the enzyme bound to the carboxylic acid portion of our peptide. This is what we call the acyl enzyme intermediate. So the second half of our reaction is going to involve breaking that acyl group off of the enzyme. And that's where the water actually comes in. So we know that this is a hydrolysis reaction, but we haven't actually seen any water yet. This is where we actually bring the water into play. So water comes in, and histidine does the exact same thing again. It's going to deprotonate water, generate the hydroxide ion right here, which will attack that carbon. And once again, we'll get the negative charge on an oxygen that stabilized by lysine. And then we're going to move the electrons back, and this time the bond that's going to be broken is the bond between the carboxylic acid and the serine residue. So this is essentially an ester hydrolysis now. And as this collapses back, our serine will take a proton away from the histidine residue, and then we will release our carboxylic acid, and then we're going to have our enzyme back in the catalytically active state that we see here. So this is a typical reaction mechanism that we would see for an enzyme that involves covalent catalysis. Basically two distinct phases to this. First, the formation of the bond between the enzyme and the substrate. The second half of the reaction, the breaking of that bond. Other things we saw histidine playing a very important role in terms of acid-base effects. We also saw electrostatic interaction playing a role between the aspartic acid residue and the histidine. The aspartic acid residue bearing a negative charge throughout the course of the reaction. That was making histidine a stronger base in this. Another enzyme that is fun to look at is alcohol dehydrogenase. In alcohol dehydrogenase, we see oxidation of alcohols. We've got a zinc cation that serves as a Lewis acid, and we use NAD+ as a coenzyme. So whereas chymotrypsin, we didn't have anything besides the actual protein playing a role in it, in alcohol dehydrogenase, we actually have a zinc cofactor and NAD+ being used as a coenzyme. Here's our zinc cation. It is complex to two cysteine residues as well as a histidine residue. And when ethanol, which is the alcohol substrate, comes into the active site, the zinc forms a complex with the hydroxyl group, the oxygen hydroxyl group, and it's going to make that a better acid. And specifically, we're going to break the oxygen to hydrogen bond. There's a base present in the enzyme that's going to deprotonate that. And as it does so, NAD+ is going to remove the hydrogen along with these two electrons. We end up with the electrons going on to the NAD+, and the electrons in the oxygen to hydrogen bond here are going to form a new carbon-oxygen double bond to make our acetaldehyde. So now we have our NADH. The NADH will leave the enzyme and go off to transfer its electrons to elsewhere. The acetaldehyde that we form will then leave the enzyme. A molecule of water comes in, and the water will deprotonate that base, and it leaves as H3O+, so we generate a little bit of acid from this as well. And then we end up with our enzyme back in the catalytically active form there. The zinc is now empty. A lot of times, if the enzyme is not doing active metabolism, we'll actually have a water molecule sit in here and complex with that zinc cation.